So hello everyone, good day everyone. The HemaTalk episode 6 on morphology is now begin. I'm Shan, the International Product Manager in Hematology line. On behalf of Mindray, I would like to say thank you and welcome all of you to be here with us today. So let me introduce our host of today's event, Dr. Javier from Spain. He is the clinical biochemistry resident at Hospital Clinic of Barcelona, and he is also the professor in the hematology module of the EFLM syllabus course, which is talk about the preparation, staining, and examination of peripheral blood and bone marrow films. Dr. Javier is very experienced in morphology, so please give him a warm welcome, and I will pass my microphone to him.
Hello.
Hello.
Hi,
okay,
hi everybody. Okay. Hi everybody, and welcome to the webinar. I am Javier, and I will be your host today. It's a great pleasure to host this event. I would like to thank to Mindray for organize this webinar and continue on me. I'm a resident in clinical biochemistry in the Hospital Clinic of Barcelona, and I will be one of the professors of the hematology module of the EFLM syllabus course. As we all know, in the laboratory, the complete blood count, known as CBC, is one of the most requested tests to allow you to have an idea of what it's like. Here in our core laboratory, we perform 330,000 of CBCs per year. As you can imagine, it is not possible to review all the peripheral blood smears and of these samples. For this reason, algorithms and analyzer alarms are useful to filter which samples to observe under the microscope. Besides, this is a time-consuming process and requires well-trained professionals. So, therefore, in recent years, we have seen many advances in this field. For example, Mindray has launched the new MC80 digital cell morphology analyzer. So, then we can see it then in a video. And this is in this slide. It's appeared to the agenda. In first place. Dr. Ana Marino will talk about the morphological detection of hematological diseases, and then we will see a video of MC80, which we can obtain more information about this new analyzer. Then Dr. Wong will talk about the importance of microscopic review and application of digital cell morphology analyzer. And finally, Dr. Olga from Poland will talk about neutrophil extracellular traps and their interaction with red blood cells and platelets. So I think that we can start. Next slide, please. Okay. Thank you. In first place, I am so glad to introduce Dr. Ana Marino. I know Ana personally, and I can say that she's an expert in blood cell morphology here in Spain. She is senior consultant in hematology in the core laboratory of the Hospital Clinic of Barcelona. She is vocal of the education committee of the International Society for Laboratory Hematology. Besides, as we can see in the slide, she has involved in more than 60 publications, focusing her research on classification and morphological recognition. For example, automatic characterization and classification of leukemic cells by means of digital image processing and pattern recognition to support the diagnosis from 2014 to 2019. So she's going to talk about morphological detection of morphological for hematological disease. So please, Dr. Anna, when you want, you can start. If someone has any question, you can write them in the chat, okay? So thank you.
So Dr. Anna, you can share your screen when you're ready. I think there is a little bit technical problems, so please be patient. Hello, Dr. Anna, are you here? Oh. Hi, everyone, just be patient. I think there is a little bit technical issue, so I am now fixing this, okay?
Now it's perfect. Thank you. I will share my screen first. Okay. Okay. First of all, I would like to thank you to Mindray for inviting me to this session. I am going to talk about the detection of hematological disease. As Dr. Laguna said, I work in the hospital clinic of Barcelona in the Core lab. So which is the contribution of the laboratory, of the hematology lab to the diagnosis? The hematology analyzer provides quantitative results of hematology parameters and flags. Blood smear morphological review led us the detection of abnormalities that cannot be detected by current automatic analyzer, such as abnormal red blood cells, presence of blasts, abnormal lymphoid cells, dysplastic signs in blood cells, platelet aggregates, abnormal platelet morphology, parasites, and so on. And it is useful, this information, for the indication of other complementary tests for the diagnosis. The modern hematology analyzers are able to quantify the different subpopulation of leukocytes circulating in blood. So the percentage of neutrophils, eosinophils, basophils, lymphocytes, monocytes, also the high fluorescent cells that correspond to immature cells, also the nucleated red blood cells, and the population of immature granulocytes. The immature granulocytes subset contains all types of immature granulocytes, such as metamyelocytes, myelocytes, and promyelocytes. Which are the warning results to pay attention when we are working in the hematology lab? First, detection of thrombocytopenia and anemia in the first blood cell analysis. In this case, a 70-year-old man is coming to the emergency department for fever and discomfort. The blood cell counts reveal a little bit high leukocyte counts, low hemoglobin, low platelet counts, and also different flags: blast, immature granulocytes, left shift, leukocytosis, anemia, and thrombocytopenia. And the dispersion diagram of the leukocytes is also abnormal. If you see the right image, this is the normal image, you can see very well the different type of leukocytes separated and different colors. In the abnormal sample, we are seeing an abnormal population on the top of the monocytes that we need to check to the microscope. This is the smear. This is a blast cell showing an immature chromatin and also a nucleolus on the nucleus. Another immature blast cell. Another one. You can see the very small cytoplasm on the top of the cell. And this is very, the shape of this nucleus is very rectangular. And this is a myeloid origin blast. Most of the myeloid blasts have this rectangular shape of the nucleus with respect to the cytoplasm. You can see the line of the nucleus is parallel to the line of the cytoplasm. This is typical of the myeloid blasts. And the diagnosis was an acute myeloid leukemia. Next case corresponds to a 86-year-old woman with a chronic renal failure in follow-up at the hospital. The blood cell counts showed normal leukocytes, low hemoglobin, and low platelet counts. And also, if you see the clusters of the cells, you can see an abnormal population similar with respect to the first case on the top of the monocytes. This was the smear showing blast cells. In this case, you can see in the nucleus the cup-like sign, morphological sign. We associate this morphology and this cup-like blast to the nucleophosmin mutation. So it's nice because there is a correlation between the morphology and the genetic. More blast cells with the cup-like on the nucleus. Here you have more. Some of them show a cleaved nucleus. You can see that the number of platelets is low. In this image, we cannot see platelets. More. So the diagnosis was an acute myeloid leukemia corresponding to an M5B monocytic Fab classification and with the nucleophosmin gene mutation. The karyotype showed trisomy 5. Next is a 53-year-old man who is coming to the hospital for ecchymosis in the last 3 weeks and epistaxis in the last 24 hours. No fever, ecchymosis in arms and legs in the clinical exploration. The blood cell counts showed a little bit low levels of leukocytes, anemia, and low platelet count. Here in the smear, you can see this big cell with an irregular nucleus profile and showing with the arrow is marked splinters. These correspond to immature granulation, and these morphological features are suggesting of that this cell is an atypical promyelocyte. You have another atypical promyelocyte showing the splinters and showing this cleaved nucleus. In occasions, it's easy to distinguish the promyelocytes, the atypical promyelocytes, because the color show a typical pink coloration in the cytoplasm. You can see the splinters also here. And this image corresponds to bone marrow. Bone marrow showing several abnormal promyelocytes with immature granulation, with splinters, and showing this pink typical coloration. Diagnosis was an acute promyelocytic leukemia with, it was demonstrated because the patient has a translocation 15-17, and also the PML RAR alpha was positive. This case corresponds to a 68-year-old woman with respiratory symptoms, myalgia, asthenia, and oral bleeding during the last month. The blood cell counts showed high number of leukocytes, low hemoglobin, low platelet counts. Flags from the analyzer were abnormal dispersion of the leukocytes diagram. You can see and compare this image with respect the normal, blast, immature granulocytes, atypical lymphocytes, left shift, leukocytosis, anemia, and thrombocytopenia. The peripheral blood smear showed again blast cells. Some of them were granulated. Some of them showed the rectangular shape that I described before for the myeloid blasts. And also, in this case, it looks like there is a cup-like morphology in this nucleus. Granulation, we can see granulation, nucleolus. Most of them are low size, but showing granulation. And the final diagnosis was acute myeloid leukemia without cytogenetical abnormalities. But some mutations in different genes were demonstrated. Nucleophosmin mutation was one of them. Next was a young man, 32 years old, coming to the emergency department after a motorbike accident. So he came to the hospital by a chance because the motorbike accident. But when we observe the blood cell counts, you can see that the hemoglobin is a little bit low and the platelet counts are also low. The graph shows an abnormal population on the left. If you compare the normal, you can see two, the lymphocytes and the monocytes, and here only there is a big cluster of cells that we don't know. So flags show an abnormal dispersion of the diagram of the leukocytes, blasts, abnormal lymphocytes, immature granulocytes, and lymphocytosis. Look the smear. We found several blast cells, low size, most of them, and the cytoplasms were practically absent. You can see two nucleolus. It was a difficult case because he came because of an accident, a motorbike accident. Also, you can see here two blast cells. They look lymphoid, this very small size. Two more. On the top, you can see nucleolus. Two nucleolus, very apparent in this blast. This is a very small, it's a difficult cell. So the diagnosis was an acute B lymphoid leukemia. It was demonstrated by the immunophenotyping of these blast cells, showing immaturity, CD34, and B monoclonal markers, but also some myeloid markers expression. The next case is a 72-year-old woman coming to the hospital for multiple vertebral fractures. The blood cell counts showed normal leukocytes, but anemia, low platelet count, and a high number of monocytes. Also, the graph was abnormal. You can see in the monocyte region a big cluster of cells. It's supposed that maybe monocytes, but we don't know. We need to check. Flags were blasts, immature granulocytes, abnormal lymphocytes, monocytosis, and anemia. Let's go to the peripheral blood smear. Oh, surprise. In the peripheral blood smear, we don't have monocytes. We have plasma cells. Yeah, we counted 54% of plasma cells and only 3% of monocytes. The diagnosis was a plasma cell leukemia. You can see here the monoclonal band on the graph, and the immunophenotype demonstrated abnormal plasma cells circulating in blood. So in the acute leukemia detection, we analyzed 187 samples with the analyzer Mindray Rbc 6800 plus, and we demonstrated that when we found low platelet count and or anemia plus flag blasts plus flag immature granulocytes, the sensitivity for the detection of acute leukemia was 94%. Second warning, detection of lymphocytosis in the first blood cell analysis. The first case was a 36-year-old man coming to the hospital because of fever, odynophagia, and discomfort during the last week. The leukocytes were normal, hemoglobin and platelets were normal, but the number of lymphocytes was high, 6.6, 10 to the 9th. Flags, abnormal dispersion of the leukocytes. You can see here in the graph that the cluster of the lymphocytes is very long, is strange. Abnormal lymphocytes, blast, and lymphocytosis. Other flags. Take a look to the smear. Oh, this is a reactive lymphoid cell because it's a big lymphocyte. The cytoplasm is very basophilic, is blue, especially on the borders of the cytoplasm when it's touching to the red cells, and the nucleus is not so mature with respect to the normal lymphocyte. We have another very similar lymphocyte, big, blue. So the diagnosis was an infectious mononucleosis, and the Epstein-Barr virus EGM was positive. Next case was a 74-year-old woman in whom a lymphocytosis is detected. Leukocytes were high, hemoglobin normal, platelets a little bit low, and lymphocytes very, very high, 66, 10 to the 9th. You can see the abnormal white blood cell dispersion on the graph with a very big cluster in the lymphocyte region, abnormal lymphocytes, flag, lymphocytosis, and leukocytosis. This is the smear. We see a lot of lymphocytes, very small, very low cytoplasm, very mature chromatin, very similar. And this chromatin is very, very typical, correspond to the abnormal lymphocytes that we see in the chronic lymphocytic leukemia. You can see this line, white lines inside the nucleus, that this morphological feature is very characteristic of the abnormal lymphocytes in CLL. So the diagnosis was a B lymphoid neoplasm, a chronic lymphocytic leukemia, and the immunophenotype showed that these lymphoid cells were positive for B, CD5, and CD23. Next case is a 30-year-old man who undergoes a medical checkup. She feels well, but she's going for a medical checkup to the doctor. And the blood cell counts showed normal leukocyte number, normal hemoglobin, normal platelet counts, but it was detected a little bit a high number of lymphocytes, but only 4.7 10 to the 9th. And the graph, the dispersion is not normal because here lymphocytes and monocytes are together. Something is wrong. The flags were blasts, abnormal lymphocytes, and lymphocytosis. Take a look to the smear. Oh, these nice cells circulating in blood is an abnormal lymphoid cell with a very round nucleus and a very special cytoplasm, because this cytoplasm is showing villus along the perimeter of all this cell. This is a typical morphology for the Hyde cells. Another Hyde cell showing this cytoplasm. This cytoplasm is not really basophilic, and is showing a lot of billows along the perimeter of the cell. Another one. Remember that this patient was for a medical check. The diagnosis was high cell leukemia with the typical immunophenotype of this disease, CD19, 20, 11, 103, 305, and 25. Next case is 59 years old man. It was positive for VIH and is coming to the hospital for skin lesions in arms. The blood cell counts showed normal leukocytes, normal hemoglobin, normal platelets, but a high number of lymphocytes, 12.8 10 to the 9th. If we look through the graph, we can see also an abnormal cluster of cells next to the lymphocytes, and the monocytes disappear. So the flags were immature granulocytes, lymphocytosis, and leukocytosis. The smear. What we are seeing here? These are big lymphoid cells, but a very particular irregular nuclear profile, and with these lines in the nucleus is a mature chromatin, but with several lines inside. This very irregular nucleus, mature chromatin, and these lines inside the nucleus is very typical of Sézary syndrome. It's a T cell lymphoma. The diagnosis was a T cell neoplasm, a Sézary syndrome, with the typical immunophenotype showing T markers and negativity for the CD7 and CD25. So our experience in the lymphoid neoplasm detection with Mindray BC 6800 plus was analyzing 45 samples from patients with lymphoid neoplasms that if we have lymphocytosis more than 4.5 10 to the 9th plus flag abnormal lymphocytes, flag lymphocytosis, the sensitivity for detection of lymphoid neoplasm was 84%. The next warning is leukocytosis and thrombocytosis detection in the first blood cell analysis. The first case corresponds to a 29 years old men coming to the hospital because of fever. The blood cell counts show a very high number of leukocytes. Look, 282, 10 to the 9th. Anemia and high counts of platelets. The graph is completely abnormal. The dispersion is nothing to see with the reality. And in the bottom, we can see this yellow population that correspond to the basophils. Flags were abnormal, leukocyte dispersion, immature granulocytes, atypical lymphocytes, lead shift, basophilia, eosinophilia, leukocytosis, thrombocytosis, and erythroblasts. Wow, a lot of immature granulocytes here, promyelocytes, myelocytes, basophils. This is a blast cell. Here we have a blast, promyelocyte, and basophil. If we have high number of leukocytes, immature granulocytes, high number of basophils, and some blasts circulating in peripheral blood, this is very suggestive of a chronic myeloid leukemia. We have precursors here. We have immature granulocytes with eosinophilic granulation. Most of them are immature granulocytes. And the diagnosis was a chronic myeloid leukemia with a 922 translocation and with BCR-ABL positive translocation. Here we have 3 chromosomes, 8, and we can see here the translocation between the 22 and the 9. 9 is here. Our experience with 81 samples from patients with myeloproliferative neoplasms was that when we found flag blasts, flag immature granulocytes, high number of basophils, basophilia, flag, or leukocytosis or thrombocytosis, the sensitivity for the detection of myeloproliferative neoplasm was 75.3%. Pancytopenia detection in the first blood cell analysis. Here we have everything is low. Leukocytes are low, platelets are low, red blood cells are low. The first case corresponds to a woman coming to the hospital because of asthenia. You can see low number of leukocytes, 2.49, 70 for hemoglobin, 34 10 to the 9 for platelets. The volume of the red blood cells was high, 101 femtoliters, and the reticulocytes are normal. Flags, pancytopenia, blasts, immature granulocytes, nucleated red blood cells, lymphopenia, leukocytopenia, anemia, and thrombocytopenia. You can see the graph is also, something is wrong here, and here something is happening. This is some images of peripheral blood cells when we can see this plastic sign in the 3 lines, because we can see here erythroblasts with basophil inclusions inside. Also, we can see here very big platelets and very strange granulation with a pseudonucleus, metamyelocyte eosinophilic, hypogranulated neutrophil. This is a ring, nucleus in a monocyte, and also this is a dysplastic neutrophil with very low granulation. So in this case, the bone marrow confirmed the diagnosis of a myelodysplastic syndrome. You can see here 2 blast cells, very the granulation of the neutrophils, more blast cells here, some precursors, red blood cell precursors also with anomalies, and the diagnosis was a myelodysplastic syndrome. Through 48 samples from patients with myelodysplastic syndromes and the analyzer Mindray Rbc 6800 plus, we see that if we have anemia and or leukopenia and or thrombocytopenia plus flag blasts plus flag immature granulocytes, the sensitivity for the detection of these syndromes was 58.3%. Which is the next challenge in the hematology lab? In my opinion, the automatic classification of blood cells. We have very good analyzers, very modern analyzers. We have the morphology. So we need, the next step is the automatic classification of these blood cells. This is just an example of the MC80 that I have the opportunity to have in the lab, showing the leukocytes, but already classified, neutrophils, lymphocytes. So this is the future, because this is, I am sure that this will improve the efficiency of our work in the lab. So let me conclude. The conclusion of this talk, I can see that the blood smear review and morphological analysis by the specialist, along with the flags provided by the analyzer, may contribute to a fast diagnosis and to select the best complementary tests useful in the differential diagnosis. And that's all. Thank you very much for your attention. And again, thank you to Mindray for the invitation to this HemaTalk webinar.
Thank you. Thank you, Dr. Ana. And thank you for the cases presented. As we have seen, peripheral blood smear review is a very important process, especially for the diagnosis and detection of hematological diseases. In addition, we have seen that analyzers can help us with their alarms. So now we shall proceed with the question time, and we have one question from Ramon Simon Lopez. He says, in adults over 45 years old, which level of lymphocytes do you recommend to provoke a smear review?
I recommend when this number is more than 4.5, 10 to the 9th. 4.5, 10 to the 9th, I think is the sign to review the smear. Okay?
Okay. He says thank you.
Thank you to you.
And well, I see that there is no more questions in the box, but I think one. I want to ask you, and in your opinion, what are the advantages of morphological analysis using digital images provided by automatic analyzers such as MC80 or television?
In my opinion, we will have a lot of advantage. using digital morphology. The quality of the cell images is very good. So we can see in a comfortable manner cells, good quality cells. So this improves the detection of, for instance, leukocyte inclusions. In my experience, using digital morphology for the automatic classification of the leukocytes, I increase the detection of leukocyte inclusions. Some examples, my Heglin anomaly. In this disease, the neutrophils show a very small inclusion, blue inclusion, and if you see the cells under the microscope, it's really difficult to see occasionally these inclusions. In contrast, with using these digital morphology analyzers, you can see all the neutrophils together, and if they have inside the cytoplasm the inclusion, it's easy for you to see this abnormality. Also, before the digital morphology, I never seen green crystals of death. These inclusions are very helpful because you need to call fast to the clinician to advise for these inclusions, because these green crystals of death in the cytoplasm of neutrophils or monocytes, it means that the liver of the patient is not working. So it's very dangerous for the patient to have that. Before the digital morphology and the possibility to see in your screen all the neutrophils, I don't remember, I didn't see these inclusions before that. More advantage. I think it is faster for the clinical pathologists to recognize abnormal cells. If you have some abnormal cells together, it's easier to recognize them with respect if you have to find these abnormal cells under the microscope, especially when the percentage of the abnormal cells circulating is low. So looking through the microscope, sometimes you can see, but maybe not. In the screen, if you have only 3, 4, but they are together, it's easier, I think, for the morphologists to recognize this. These are summarizing some of the advantage. I think the efficiency of the hematology lab will be better using these digital morphology analyzers.
Okay, well, now we have 4 questions more. The first one from Ms. Warfata, it's a question about standardization. He says, how to standardization combination in interpretation by specialists and advanced hematology analyzer?
I think that if you have an advanced hematology analyzer and an experimented morphologist, the work will be perfect. The combination. I think people working in morphology, we have usually protocols in our labs for standardize when we have to review. I think each lab have to standardize this protocol. The ICSH organization published some years ago some rules for the standardization. It is obligated, the standardization of these rules in the hematology lab.
Okay, the next one, Justina, she says, in television, erythrocytes and platelets may not be well seen, and some problems on their morphology may be overseen, I think. Well,
I think in my experience, erythrocytes, I don't have any problem to see the erythrocytes in the screen using CellaVision, okay? Because the only problem is that you need to have a very good staining. If the staining is not good, you are not going to see red cell abnormalities on CellaVision, on the microscope, on Ep6, 80, no. See, the first rule is obligated and staining perfect. If the staining is perfect, I think that you can see very well the morphology using the CellaVision. I don't know, because you can look parasites, you can look inclusions, different abnormal cells. The only thing that my experience, the CellaVisions, I know CellaVision because usually I work with CellaVision. The analyzer said, for instance, parasites, and you look through the smear and no, because there are a lot of false positives for parasites using CellaVision and other abnormalities. You need to check the morphology. The software for the red blood cell analysis is not perfect, but using the images provided by the CellaVision, I don't have any problem to analyze. With respect to the platelets, is different because in some cases you need to check if maybe there are some aggregates in a part of the smear, and maybe the CellaVision is not taking the pictures of these aggregates. But you can suspect that if you check and you cannot relate the number of platelets with the pictures that you are seeing. You need to take care, but in my opinion, it's quite good on red blood cells, and occasionally you need to check the smear under the microscope to see if there are aggregates or not.
Okay, Ramon Simon says, do you think that allow NEX NE site scatter dispersion may help to improve the detection of myelodysplastic syndromes?
Maybe yes, because the neutrophils in the myelodysplastic syndromes are not normal. They show low granularity, and in some cases, they don't show any granulation. So this, I think, can be detected using scatter dispersion for the neutrophils. I think it's a good idea to check.
Okay, Kaushik asks, should every morphological diagnosis be confirmed with the immunophenotyping?
Everything not. Everything not. For instance, if you see Plasmodium, no, and this is a morphological finding. But with respect to blast, with respect to abnormal lymphoid cells, yes. If you need to prove that these cells are blast, and these blasts are from myeloid or lymphoid origin. Is obligate to do the immunophenotyping for the diagnosis of leukemias and lymphomas, but other not. If you have reactive lymphocytes, you are not going to do immunophenotype. You need to check if you have a virus infection, but not immunophenotype. For instance, in a promyelocytic leukemia, it's more important to demonstrate the 15-17 translocation. for the diagnosis, more than the immunophenotyping. Okay?
Perfect. And the last question, a 45-year-old male patient has a lymphadenopathy in inguinal, a slight increase of leukocyte, and from a smear I found immature neutrophils and some abnormal nucleus that I can distinguish whether it's a lymphocyte or immature granulocyte. Should I curious for cancer?
A veure, immature neutrophil, impossible. The neutrophil is mature. You cannot say immature neutrophil. Okay? The neutrophil is a mature cell. Immature cells, if you see immature cells, abnormal nucleus, of course, you need to check if something is wrong with this patient. Morphology can alert, if you are not confident that the cells that you are seeing are not normal, you need to check. But take care, because the neutrophil, the chromatin is mature in all the occasions.
There is one more question, the last one.
Yes.
Does automated smear analysis systems accurate count abnormal cells and young forms?
Quite good. They are quite good, yeah. When they alert for a immature granulocyte population, usually you find left sheath and immature granulocytes. I think, yeah, it's quite good with respect to this question. Thank you very much for all these questions and to share with me this presentation. I hope you like it.
Okay, thank you, Dr. Anna, for your session. And now we are going to watch a video about the new hematology automated analyzer. As we commented before, Mindray has launched the new MC80 digital cell morphology analyzer. And in my opinion, MC8 takes digital morphological analysis to the next level, and offering clearer images that can capture abnormalities in greater details. So let's have a look. Well, it's incredible. I think it's a great technological advance in the future of laboratory hematology. And then with regard to the video, we have the presence of Dr. Wong. He works in Shanghai Center for Clinical Laboratory. In addition, he is associate professor at Shanghai Jiao Tong University School of Medicine. He is professor at Shanghai University of Medicine and Health Science and auditor for the ESO 15189. He's an expert for procurement department of Shanghai government, and he has published over 30 journals and obtained one national technology invasion patent. He's going to talk about the importance of microscopic review and the application of digital cell morphology analyzer. So please, Dr. Wong, when you want.
各位专家、各位同行，上午好。态学分析技术。
Today I'm going to share with you the importance of microscopic review and application of digital cell morphology.
60年代
We all know that in 1960s, and we already started to see cell scan, HemaTalk, and cytodoc scanning.
一些这个仪器，那么包括这个我们白细胞的自动测备的计算机，还有库尔特的 DIF3 和 DIF4，
and also the automatic WBC counter and DIF3 and DIF4 BCI, as well as ADC 500 produced by Abbott. And later, we started to see the flow-based cell counter in 1980s.
到了现在呢，已经我们
Well,
有
nowadays,
显微成像的这个设备
we
系统
have the automatic digital microscopic recognition and the imaging system.
那么这个自动的数字细胞形态分析仪器呢，有
Well, for this digital cell morphology system, it actually have the following features. On one side, it is a microscope. It also has camera and analysis system with software.
数字细胞形态学分析仪
So you can say that nowadays, this digital cell morphology analyzer
这个 Micro 21，还有
and
DM
including Micro 21, the intelligent medical imaging, Palm Beach Gardens and FL, and also the DM96, DM1200, and DM 9600, the salivation AB. We also have the DI60, the SysMac and cleavation one, designed and developed based upon DM 1200.
80。那么当然，这个到目前为止，大多
So
数
you
的
can
这
say
个
that nowadays, majority of those devices
是完成的。那么自动的数字细胞
So
形态
now,
学
if we take a look at the digital cell morphology analyzer, you can say that they mainly include the following workflows, including the sample collection and around 20% of the review rate, and the peripheral blood smear and staining, also the barcode preparation, and we put it into the slide box and put the slide box into the analyzer, and then we do the pre-classifications. And I like to mention that peripheral blood smear and staining is quite important, the same as barcode preparation. And the final step, pre-classification, is also quite important.
我们自动的数字细胞形态分析仪呢，它的
So for this digital cell morphology analyzer, we can say that it actually provides very good principles of the pre-classifications. And for example, it can actually base of the below 6 characteristics, including the shape, the color, the texture, the wavelengths, the Markov, and the wavelets, and then to characterize the cells according to the pre-classification.
这个中性粒细胞的分异盒淋巴细胞单核细
And
胞
also the
等等。
EO as well as the base Q.
我们大家可以看到，我们这个自动的一分类这个它的显示的界面，它这个
And let's now take a look at this digital cell morphology analyzer, including the pre-classification and the manual review. And we have the interface from Mindray newly launched digital cell morphology analyzer, the MC80. It's a Chinese version.
我们 DM96 这个
And also we have DM96 And DM96 is actually a WBC pre-classification. It can be used for 120 blood samples that triggered the re-examination rows. And the result from the automatic digital cell analyzer is 82% consistent with the manual microscopic review. The consistency of mature cell is higher than the immature and abnormal cells. The low consistency of the eosinophils and the basophils is related to the small number of the cells. And also, it shows great advantage of the digital cell analyzer. It can do remote review confirmation and reduce the workload of the staffs. So you can say that this is the general pre-classifications we have. Well, regarding the digital cell morphology analyzer, the DM peer review, you can see the correlation between digital cell morphology analyzer and the manual microscopic review is good. And also, the lymph and the big blaster have a better correlation. And also, it has a relatively poor correlation between the basophils and the prominent site, probably due to the low quantity. But we also clearly notice the stage correlation of the immature cells, I mean the early, mid, and late immature cells, is lower than that of mature cells. So we see a very good correlation between the immunogranular site. This is also.
总体来说，我们的幼稚细胞、不成熟细胞的这个相关性呢，还是比较
You
好
can
的。
say that generally speaking, we see good correlation between immunogranular site.
我们再来看看我们这个
Well, let's also now take a look at the digital cell morphology analyzer, the DM peer review.
这个链型红细胞和这个口径红细胞，
Let's
它的这
see
个
the Rbc characterization. First of all, the left side shows the example screenshot from the Standardization DM96 system showing the normal red blood cell. You can see very high sensitivity and specificity for the sickle cells and the stromocyte, and also poor sensitivities for the bar cell and Rbc agglutination.
那么对于异常红细胞的这个设备情况，我们可以看这是一个骨髓纤维化患者。
This is a patient with the typical teardrop cells in the blood smear of a patient with myelofibrosis. And you can see this pre-classification by the DM96 has a relatively good correlation between the manual microscopic review and also after the DM analysis, and the proportion of the teardrop cell in normal person is less than 0.5%, where for the patient is always more than 0.5%. The digital cell analyzer helps to pre-classify the cell quickly.
通常是要高于0.5%的。那么我们自动阅片机
Well, in this way, you can see that the automatic reader can actually provide a better view in this regard.
呢，大家可以看这张图跟表啊。那么我们对于127例的外周血样本，那么我们用这个血液分析，
You can say that we use the
进行这个比较。那么
cell
结果呢，
morphology analyzer, the DM, for the peer review, especially for platelet recognition. And the sample size is around 127 peripheral blood sample. And the methodology is that the hematology analyzer and the manual microscopic review and the automatic digital cell analyzer. The summary is that the correlation between manual count and the CVDM96 is good with and the automatic hematology analyzer result with CVDM96 is good.
那么
So we can generally see that the sensitivity of the PLT clump detection is poor, and it needs manual review on smear to improve the detection rate.
敏感度啊，大家可以看。
This is generally what we have in this study, and you can also see that.
8.8。那么我们随着这个不同的方法介入以后呢，我们的灵敏度逐步的提升啊。那么我们这个常规在阅片的时候呢，只能
So you can say that originally speaking, that when we are reading the smears, and what we're trying to do is to see and make sure that smears is readable with a feather edge and the readable area of more than 100% of the PLT clumps being detected.
阅片区加上这个 双侧波边的双侧，再加上尾部，那么可以实现这个100%的检测到血小板的聚集。那么另外一些其他的特殊形态的识别，我们看看这个我们的这个对于这个疟原虫它的检测情况。那么这是在这个我们的133疟原虫。
You can also take a look at the special cell recognition. We have a 130 plasmodial and polyparium infected red blood cells, and the B cell positive cells, and 151 negative cells for double-blind detection under DM96. On the left side, you can see the PF captured by the DM96 on the Rbc detection. On the right side, you can see the PF at the high magnification conventional light microscopy. And if you take a look at the result, you can see that the CV plus pathologics review, the accuracy is around 85%, where the sensitivity for CV plus technologies review for a detection of the intracellular was around 73%. Let's now also take a look at the digital cell morphology analyzer, the DM. The correlation of automatic cell morphology analyzer with manual microscopic review for 149 patients, and we find out the pre-classification of the cell for the CV is relatively low, except neutrophils, but CV plus manual review improved the accuracy, and the accuracy of the WBC pre-classification has no correlation to HIV infection status. The accuracy is acceptable for CV plus manual review, with the price of CV is relatively higher for South Africa. And you can also see that the rate is being greatly improved. So we can see that the recognition of the cell for the cancer center, and clearly you can see for some the bound neutrophils, the sensitivity is around 74.57%. And this is a recognition of the cell at the Children's Hospital, and also the band neutrophil is around 74.46% for the sensitivity and 97.25 for the specificity. Where at the same time, we can also clearly notice that for basophil, the sensitivity is 85.45%, while specificity is 99.83%. So we can say that actually it shows very good recognition of the cells with very high sensitivity from the basophil and also from the lymphocyte, and also its specificity are always be more than 95%. And we can also clearly say that for other cells, and especially if we place this study into a children's hospital, and also the band neutrophilia, especially for the DM, according to the peer review, and the sensitivity is being also improved on the segmented neutrophils. And also for the primer locyte, it can reach 100%. So you can clearly see that for the blast and also the sensitivity is more than 90%, where for specificity, actually over all the blue dotted area, it's always more than 99%. Let's also take a look at the improvement of the analysis time. You can see that the throughput of the WBC pre-classification is being greatly improved. It could reach 30 slides per hour, and also smaller difference in analysis time for DM96 with reclassification of cells, and also larger difference in analyzed time for manual differential analysis.
看到啊，我们可以从1小时
especially within 1 hour, you can clearly see the throughput of WPC pre-classifications being greatly improved. So this is a great improvement made upon the existing technology, and we are happy to show this result to all of you. On the left side, we show you the table of the comparison of the time taken to complete the 30 differentiation on the cellular vision DM96. Coming next, let's also take a look at these digital cell morphology analyzers' advantages and disadvantages, including the advantages include actually it enable remote review, but approximately 10 to 20% of the slides may still need manual review. The digital archiving, it's easy retrieval of the blood films, and it can reduce fatigue and eye strains. It can also reduce labor cost, but we still need skilled labor for this work. It can also facilitate the morphology education, QA training, and peer review in digital format.
80以上的这个，我们可以通过这个过筛。
So you can say that this is truly an advanced technology for people to use. Where on the right side, we should also take a look at the disadvantage. Well, from the disadvantage perspective, we say that all samples from the newborns and from the patients with leukemia and suspicion of the presence of the pathological cytotypes, including the blasts, the plasma cell, and the immature granulocyte, suspected the plastic cells, suspected others, and also the screening of the intracellular parasites. And there's another targeted 81 hospitals using DM, and we see the feedback from the 37 hospitals. We can already clearly see the difference being seen over there. The advantage is that it can help to reduce visual fatigue, and it can also result in a relatively high consistent result. Where at the same time, we can also facilitate the personal training and also the Rbc and the PRT count is also a disadvantage, and 35.7% of the feedback saying it is not suitable for neonatal blood smear. In addition, the reason why 8 users did not use DM, I think the reason is because some hospitals believe it's too expensive, and also the difference between the image taken by DI and what you see under the microscope is still relatively large, and also you can see less review samples, and also TAT time is also a great concern for some of the users why don't come to choose to use DM. This is what we noticed from the market and also from the question now. Let's also take a look at other digital cell morphology system, including the interface for WBC and also the interface for RBC and PRT. For next slide, it has also a good throughput. It can reach 26 slides per hour, and also we collected 4,479 samples. The correlation is good compared to Menu's microscopic review, and it relatively correlation for basophils and bands due to the low quantity. And let's also take a look at the next slide. You can see that compared with DM96, it shows good correlation for all cell types, except basal cells and immature granulocytes. Let's also take a look at the ICSH recommendation. According to the recommendation, we can see result has to be consistent with the hematology analyzer and the flex to avoid the missing diagnosis. And especially from this table, we can see that digital morphology analyzer in hematology is being recommended made by ICSH review and recommendation in 2019. Where at the same time, we need to pay more attention to the difference between the blood smear stand by automatic slide machine and the blood smear stand manually. The blood smell stained manually may look large, thinner, and the color difference may also occur. And it may also lead to misclassification of the abnormal cells and the lymphocytes into the blast.
和颜色的差异呢，可能导致细胞识别的不正确，进而导致原始细胞的假性偏高。所以
And
这
it
方面也是
also
要。
lead to some of the
另外这
issues
个
we may clearly notice in our practice. Another part is
目前
on
我们
the standardization of the morphological analysis. And the problems that we are facing for standardization is that we still need to do more standardization in sample preparations, staining procedures, the hardwares, including the automatic slide maker and the softwares. Where on the right side, we show you the standardized documents for digital cell morphology analyzer.
还有一些
For
这个国际
example, CPA published the guidelines for verification of smear in pathology. FDA and ICC co-hosted a morphological
那么
cell
我们
analysis
对于这个
forum. And we can also say that from the practical perspective, we made the following recommendation for morphology experts and groups. Standardization of the color of the image for easy communication of results and information sharing. And we should also have the quality of the digital image based upon their usefulness of the pathologist, instead of pursuing whether the image looks compared to the microscopic or not.
直观的观察的
We should
更好的
also
更差，因为
try to
在我们 DM
establish
系统
a mechanism to objectively evaluate the quality of the image.
它只要能够分出就可以。那么另外，是不是建立一种机制，
Where on
可以
the
客观的评价？
As I mentioned, we need to establish a mechanism to be more objective in evaluating the quality of the
对于
image.
我们自动电梯的厂
Well,
商
I
的
can
厂
say
家，
that for social groups, we should also support the pathologists in setting the standard.
那么目前这个 DM 呢，可能用的是60
I
倍、
think
50倍的邮径啊。
For
另外，不同的
DM,
用
actually, different manufacturers, they're probably going to use different oil lengths. And the database for the cell image can be shared by different manufacturers. And I think the deviation during the staining process can also be calibrated by the analyzer. It can help to improve the accuracy of WBC, RBC, and abnormal cell pre-classification. It can help to update the Rbc characterization according to the updated guidelines.
另外，从一篇分学分
It
析
can
的结
also
果让
help
有
to
机的。
integrate the result from a hematology analyzer and
那么对
digital
于我
cell
们使用这
morphology.
个月编辑的实验，
So according to
也
that,
是
we also have some recommendation to labs.
首先要做好的就是
First of
目
all,
前用的人工镜
you need to
跟我
set
们
up
的
a methodology mostly with manual microscopic review to compare with the automatic digital morphology analyzer. You at least need to have 50 blood smells, including all cell types and abnormal cells. And it should also extremely important is that if a lab is going to switch from a manuscopic review to automatic slide maker, you need to guarantee you have some blood smells with all blood types and abnormal cells. And also at the same time, we need to be proactively embrace the transformation from a manual microscopic review to digital cell morphology analyzer. And the manufacturer should familiarize you the difference between manual microscopic review and automatic digital cell morphology analyzer. Share more high quality smell for reference. It be useful for local regulatory authorities for EQA sample preparation. The labs should also put the blood smear analysis into the first priority of the clinical diagnosis, therefore to reduce the morphology experts' workload. In this way, it will help us to further reduce workload for the team. For example, we can focus on the abnormal cells and label them for future analysis. In 2016, Searle put stomatocyte and steatotype and the teardrop cells and spherical shaped RBC and the parasite as a special label for further analysis. My conclusion is that let's embrace the digital cell morphology analysis. Let's set up standardized guidelines. Let's also try to improve the accuracy of a pre-classification of the cells. Let's also focus on the abnormal cells. And we should also familiarize with the difference between manual microscopic review and the automatic digital cell morphology system. And we should also be familiar with the advantage of the limitation of the digital cell morphological analyzer so that we can integrate the result from a hematology analysis and the digital cell morphology system as a mutual verification. Only in this way, it can help us to further improve the quality of our review in clinical settings.
细胞呢进行这个标记啊，使得这个我们这个分类的时候呢，能够这个准确的分类。那么另外还有熟悉人工显微镜观察和自动型的这个阅片它的差异。
And I mentioned that we need to clearly understand the difference from the manual microscopic review and the automatic digital cell morphology system to help to translate the disadvantage into advantage.
临床这个形态学人员解放我们的这个
And also, we could be unburdened from all the heavily manual microscopic reviews, so that we can spare some manpower for those review that truly need the manual power.
这个我们的小方图啊、散点图啊，还有一些报警的信号，那么一定要。
And we can also notice some of the norms as well as the digital information into this to make sure that we have a systematic development in this regard.
给到的一些这个对于这个自动数字形态学分析的这方面的一些总结。那么我要分享的内容呢，大概就到这里啊，谢谢大家。
That's all for me. Thank you.
Well, thank you, Dr. Wong, for your presentation. Can you hear me? Yeah, okay. I agree with you. Laboratories should put blood smear analysis into the first priority for clinical diagnosis. And as we have seen in the webinar, a laboratory professional can provide a lot of information just with a hematological analysis and peripheral blood smear review. So we shall proceed to question time. If someone has a question, please leave it in the question and answer box. I see that there is. Okay. Well, I see that there is no question in the box. So I'll start. Professor Wong, what do you think about the advantages of the digital cell morphological analyzers used in a clinical laboratory? Hello?
Hello?
Dr. Javier, would you mind to repeat the questions?
I can hear you.
Dr. Javier, would you mind to repeat the question? Because Dr. Wong cannot hear about the question.
Okay, I asked about the advantages of the use of digital cell morphological analyzers in a clinical laboratory.
Okay, I will explain. I will translate to Dr. Wong. Please wait a moment.
Okay.
它的优点就是那个能够集中我们的
Its advantage is that it will be able to do the remote review and also make sure that the morphological expert could be free from the heavy clinical burden where at the same time, it can also be used and to reduce the fatigue, the eye strains, and it can also be used for educational purposes. It can also help to reduce the fatigue. So I think this is a great advantage of the digital imaging-based system. But for sure, it also has some disadvantage. Especially, you can say that and it's still somewhat different from the microscopic manual review. For example, it has different the lenses. It used to adopt the 51, where for MCAT, I think for the oil lens, and it's around 100 times. So it's more applicable for clinical settings, especially for the morphology analyzer. So I think that's a advantage of the disadvantage of the digital cell morphology analyzer. The oil lens is quite differentiated from the microscopic manual review, where I think a lot of evaluations been made on CELOV, where for Mindray's MCAT, it's only been released recently, so we don't see any kind of assessment now, but I think this is a good product.
Okay. If someone has a question, please leave it in the question and answers box. So if there is no more questions, okay, well, question time is finished, and thank you so much, Dr. Wong, for your presentation.
Thank you.
So finally, we have Dr. Olga Ciepiela. She's going to talk about neutrophil extracellular traps. She is the head of the laboratory medicine department in the Warsaw University of Medicine, and she is the head of central laboratory in the UCK. She's member of the board of Polish Society for Laboratory Medicine, and she has published over 70 publications in the field of laboratory medicine, hematology, and immunology. Well, she has been the principal investigator or co-investigator in 8 research grants in the field of neutrophil extracellular traps studies. So please, Dr. Ciepiela, you can start when you want.
Javier, thank you very much for the introduction. I'm really impressed how you pronounced my name. It was very good. Thank you very much. Hello, everyone. My name is Olga Ciepiela, and as Javier said, I'm an associate professor at the Medical University of Warsaw, and I also work as the head of central laboratory at the University Clinical Center in Warsaw. In the beginning, I would like to thank Mindray for inviting me to this event. I'm honored to have the possibility to share with you my scientific interests. Through my whole scientific life, I was involved in the studies of hematology and immunology, but unfortunately, these topics were always next to each other. My main subject of studies in the field of hematology are the red blood cells and hemolytic anemias, mainly the hereditary spherocytosis. Several years ago, I started research in the field of innate immunity. I was, and I'm still interested in the mechanism in which neutrophils protect our body against infection. Oh, sorry, I didn't share the screen. Here, you didn't tell me. Sure. One more time, sorry. Can you see the presentation? No?
Not in full screen, not.
Click the display. Yeah.
I was trying to. I knew it will happen.
Just share the screen and display. We can see your screen just now. but not now. I mean,
can you see it now?
No, not yet. You can close it and.
Yes, I will close it. Please wait a minute.
Yeah, sure. Take your time. Okay.
One more time.
Okay. Click the display. Yeah, play. Thank you.
Is it okay? Can you see it right now?
Yes.
Yeah.
Yes,
it's
great.
working.
Thank you very much. I knew it happens. Okay, so thanks to my previous team, I started research in the field of neutrophil extracellular traps. And today I will tell you something about these traps and their interaction with red blood cells and platelets. So let's go to the point. In March 2004, in Science, the Science published a paper of Professor Arturo Rzechliński and Dr. Volker Brinkmann. This paper was titled Neutrophil Extracellular Traps. And actually, that was the beginning. Neutrophil extracellular traps kill bacteria. That was the beginning of the studies in neutrophil extracellular traps. The team from Max Planck Institute showed that neutrophils can release threads of DNA which are decorated with some proteins, bactericidal proteins from cell granulae from inside the cell. These threads were named extracellular traps because they were able to entrap and immobilize the bacteria, the pathogens. These fibers were found to be released upon cell activation with either gram-positive or gram-negative bacteria. They also showed that pathogens are not only immobilized within these traps, but they are also killed and their virulence factors are degraded. That was one of the first reports showing that nets may play a very important role in innate immunity. They made a high local concentration of antimicrobial agents to kill bacteria. Volker Brinkmann is the author of this absolutely beautiful picture made with use of electron microscopy, in which we can see the sticky threads of DNA and the bacteria which are entrapped within the nets. I couldn't help myself not to show you how Pedromics imagines the neutrophil extracellular trap. So in this picture on the right, you can see that we already have Spider-Mans and they are really alive, right? But let's go back to the nets. These antimicrobial structures are composed of DNA complexed with histones, as well as granular and cytoplasmic proteins, such as neutrophil elastase, myeloperoxidase, and cathepsin G. Actually, we already know over 700 different proteins that are found in the net structures, and their sources are cytoplasm, granulae, nucleus, but also the mitochondrion. Why mitochondrion? Most studies point to nucleus as the source of nets-associated DNA. However, the release of mitochondrial DNA has also been described in the net structure. This phenomenon is associated with so-called non-lytic net release. Very often we can meet the term netosis, which underlines that cells after releasing nets die by the suicidal death. However, it has been also shown that neutrophils can live for some time without nucleus, still phagocytizing, so we can't say that they undergo netosis because they are still living. They just released nets. Cell living without nucleus is nothing special for us, especially as a hematologist, because we know red blood cells which live over 120 days and they very well play their role. Release of nets composed of nuclear DNA is not the rapid mechanism. It needs even 2-3 hours to happen. But another studies showed that some nets are released even within tenths of seconds, so very, very quickly. And thanks to the staining, specific staining, it was shown that these nets, these very rapid nets, are formed of the mitochondrial DNA. As we already told, in physiological conditions, these DNA-containing traps immobilize and kill microorganisms, but not only bacteria. Their release was also found in parasitic, fungal, and viral infections. Interestingly, neutrophils seem to sense the size of the pathogen, and they can choose if they use the phagocytosis or release of neutrophil extracellular traps. Mechanism of NETs release is quite complex and involves several pathways. During this process, we can see that nucleus decondensates and intracellular membranes disintegrate, allowing the mixing of nuclear and cytoplasmic components. Eventually, the plasma membrane is disrupted and the nets are released outside the cell. There are over 300 compounds that trigger cells to release nets. In addition to microorganisms, nets are triggered by other various stimuli, including pro-inflammatory cytokines like interleukin-8, like tumor necrosis factor alpha, platelets, activated endothelial cells, nitric oxide, monosodium urate crystals, various autoantibodies, and many others. These triggers cause neutrophils to activate kinases that induce assembly of an enzyme complex called NADPH oxidase, DNOX. NOx then produces large amounts of superoxide, a highly reactive oxygen species that carries an extra electron during a process called the neutrophil oxidative burst. Reactive oxygen species resulting from the oxidative burst trigger disintegration of multiprotein complex to release active neutrophil elastase, which is primary component of NETs. And this neutrophil elastase is released into the cytoplasm. Then the neutrophil elastase migrates to the nucleus where it cleaves histones and other proteins to decondense the chromatin. As you can see in the right picture, these are the pictures from our study. Different compounds can cause migration of neutrophil elastase into the nucleus. Here we can see the green parts of in the merged picture, the green parts of the elastase inside the nucleus. Another stage of NETs release is citrullination of histones, and citrullinated histones H3 and H4 were found to be typical markers of NETs release. Histone deamination by peptidyl arginine deaminase, so the enzyme called PAD4, is a prominent post-translation modification in NETs that is induced by inflammatory state. The conversion of certain arginine residues to citrulline in core histones by PAD4 eliminates the positive charge from the residue and may thereby weaken the binding of histones to DNA. So eventually, as a fact, the chromatin fills up the entire cell until the cell lyses and extrudes nets into the extracellular space. So first, we can see the decondensation of chromatin, migration of neutrophil elastase into the nucleus, then the release of nets outside the cell. So we already know that nets is a mechanism of innate immunity that protects us against infection. Unfortunately, nets are also recognized as double-edged sword of innate immunity. Why we said the double-edged sword? Since nets contain a magnitude of proteolytically active proteins, an uncontrolled nets formation process may promote tissue damage. Imbalance between nets formation and clearance can contribute to multiple pathological conditions such as systemic lupus erythematosus, thrombosis, cystic fibrosis, or even formation of cancer metastasis. Several of the molecules decorating the nets, for example, myeloperoxidase or double-stranded DNA or histones, are autoantigens in systemic autoimmune diseases. Various groups have proposed that aberrant net formation may be important in the generation of autoimmune responses in predisposed individuals. This hypothesis is supported by the observation that initiation or exacerbation of autoimmune diseases often occurs following microbial infections. Role of NETs in cancer is also complex and mutual. This includes, on the one hand, forming a physical barrier between cancer cells and immune competent cells and inhibiting the functions of other immune cells, as well as also promoting migration of cancer cells because they are entrapped into the nets. On the other hand, nets can kill the cancer cells and stimulate the immune system by activating T cells, NK cells, or macrophages. They also can recruit other neutrophils to the place of tumor and help to fight with the tumor. We have already mentioned that neutrophil extracellular traps contribute to thrombosis. The way in which nets promote coagulation is very complex and involves the activation of either platelets or plasma coagulation factors. It has been shown that nets themselves can induce and activate coagulation factor 12, simultaneously inhibiting the tissue factor pathway inhibitor, TFPI, by serine proteases, together promoting coagulation. Furthermore, a negatively charged DNA found in the net structure was found as a factor that activates intrinsic pathway of coagulation. Moreover, both tissue factor, so the TF and extrinsic pathway part, and von Willebrand factor have been observed in the net's composition, together with coagulation factor 10, prothrombin, fibrinogen, leading to the suspicion that nets trigger coagulation by affecting either intrinsic or extrinsic pathway of coagulation. Direct evidences of nets engagement in thrombi generation was also provided. The immunohistochemistry staining showed in the thrombi the neutrophil elastase, the myeloperoxidase, the DNA, the citrulinated histones. And also it was found that DNases, so the enzymes that can digest DNA, help to lyse the thrombi. So we have the evidence that neutrophil extracellular traps contribute to thrombosis. As I mentioned, nets and also interacts with platelets, and this interplay is mutual. So we know that nets can trigger coagulation, but we also found that platelets can activate neutrophils to release neutrophil extracellular traps. It has been shown that platelets and platelets-derived factors like high mobility group box 1 or p-selectin are able to activate neutrophils and make them to release nets. In contrast, nets and their components activate platelets mainly by histones H3 and H4, but also by histone and DNA complexes through stimulation of toll-like receptors TLR2 and TLR4 on the surface of platelets. Due to its sticky character, DNA from nets becomes a scaffold for the thrombus generation, and its negative charge causes activation and aggregation of platelets. Nets' effect on aggregation and platelets activation was confirmed by release of ATP and ADP from platelets, an increase of surface expression of CD62 ligand, and phosphatidylserine in the presence of net fibers. Interestingly, no DNA or histones are involved in platelet aggregation, but cathepsin G found in a structure of nets and phosphorylation of 6-tyrosine kinase in platelets. Regardless the mechanism, an effect of nets on platelets results in formation of thrombi, either venous or arterial. This pathology was observed in acute inflammatory states, but also in cancers. So these were the platelets. What about the red blood cells? As we know, they are also involved in thrombus generation, as they can be trapped within fibrin fibers and clot. when studying NET's effect on thrombus generation, it has been found that negative charge of DNA may also entrap red blood cells in NETs-based thrombi. So, as you can see in this picture, NETs become a scaffold for platelets, but also for erythrocytes that are trapped in one thrombus. We have to take into consideration that red blood cells which are entrapped in the neutrophil extracellular traps may be damaged and they may hemolyze. And hemolyzing red blood cells may also promote neutrophil extracellular traps release. Several groups ask the question if the hem or hemolysis can affect NET release. So the answer is quite clear. It's yes, because in the study with a sickle cell disease subject, it was shown that hem itself can induce neutrophil extracellular traps release from intact neutrophils. But also the hem is able to stimulate the neutrophils which are primed with inflammatory cytokines in the beginning. In this picture from the paper of Cheng et al., we can see that hem triggers release of NETs, and adding the DNases causes that neutrophil extracellular traps are no more present in the field. So this is the direct evidence that hem itself can promote and can stimulate neutrophil extracellular traps. As we know, sickle cell disease symptoms are associated with vaso-occlusive crisis. A vaso-occlusive crisis occurs when the microcirculation is obstructed by sickle red blood cells, causing ischemic injury to the organ supplied and resultant pain. But involvement of NETs in the pathogenesis of thrombus generation, as well as ability of DNA to bind and aggregate red blood cells, shed a new light on the causes of vaso-occlusive crisis in the course of sickle cell disease. As you can see in these pictures, hem released from sickle cells induced a release of NETs, and then other red blood cells get trapped into the NETs. My studies have shown that treatment of sickle cell disease patients not only increases soluble fragments of NETs, so the free DNA, but also may possibly protect against this vaso-occlusive crisis. So far, we know that DNase, the enzyme that digests DNA, decreases a risk of acute lung injury in mice, at the same time prolonging their life. So maybe this is the partially treatment for the vaso-occlusive crisis in patients with sickle cell disease. To summarize NET and sickle cell disease, or maybe every hemolytic anemia, it can be said that hemolysis of the sickle red blood cells produces hem microparticles, which activates neutrophils to release chromatin and granule proteins to form neutrophil extracellular traps. So how we can see in this picture, here we have this thrombus with involved NETs, neutrophils, platelets, and sickle red blood cells. Induction of NETs by hem indicates that red blood cells in some way may be the part of innate immunity. This a little bit disrupts our previous belief that red blood cells are just naive immune cells and they just transport oxygen. But involvement of red blood cells in NET mechanism is just a beginning. Recent studies have shown that red blood cells can bind DNA from pathogens, and in that way, they can induce the immunity response. One important immunomodulatory property of human erythrocytes is their ability to bind a wide variety of chemokines. They are bound to the Duffy antigen receptor for chemokines on the red blood cells. Recent work has shown also the ability of erythrocytes to bind the mitochondrial DNA. And as we already know, the mitochondrial DNA can be found in the NET structure. So This is another point that neutrophil extracellular traps can be bound to the red blood cells. It has also been found that pathogen-derived DNA may be bound to the toll-like receptor 9 on the surface of erythrocytes. Then they are erythrophagocytes, and next they promote innate immune response. Erythrocytes can also directly bind pathogens. The best known one is the Plasmodium, which is bound to the red blood cell glycophorin A, B, and C. So to summarize, in our clinical laboratories, we very often analyze the number and the morphology of platelets. We also analyze the number, the indices, and the morphology of red blood cells. In our daily routine, we do not realize that the cells that we observe in the blood smear, and with which we are very often in love, of course, as a hematologist, interplay with each other, thus influencing our innate immunity. I hope that next time you will see the red blood cell and the platelets in the blood smear, you'll think more warmly about these cells. Thank you very much for your attention.
Well, thank you, Dr. Ciepila. In my opinion, I think it is a very interesting presentation. Personally, I was not aware of the existence of nets, and I think that when we are asked about a function of neutrophils, we always think in phagocytosis. Although we know that the professionals of phagocytosis are the monocytes. So thank you for teaching us something new, at least for me. And now we shall proceed to the question time. We have 2 questions in the box. The first one says, so in your conclusion, the elevation of nets is good or bad prognosis in infections, and are we need to check for nets in all infection or just in certain conditions?
I think it's a bad prognosis in severe infections like sepsis. Several studies has shown that increase of citrullinated histone H3 in patients with sepsis is associated with the bad prognosis. So if you have the patient with severe infection and we have increased markers of net release, we can worry about these patients because the state is really severe and acute.
Okay. Next question. Does NET contribute to disseminated intravascular coagulation and organ damage in sepsis?
Absolutely, yes. It has been found that in DIC, this thrombi that are formed in the beginning of the process also include nets. So the release of nets is because we can remember that in DIC we have this total cytokine storm, and this cytokine storm can also stimulate neutrophils to release nets, and then nets bind to the thrombi, to the red blood cells, to the activated platelets, and they can be found in a different organ and they can damage them. So it's absolutely yes.
Okay. I see that there is no more question. I had one question. When I read the title of the presentation, I looked forward about nets, and I read a paper from your research group, and I want you to ask something about the role of zinc in these nets.
Of what?
Zinc.
Can you write it, please, in chat box? It will be easier. Zinc. Okay. Yes, I get it. It's our last paper published in Nutrients. We found recently that zinc is important in the release of nets, and we also found that exert of zincas can affect the release of nets, causing that the nets that the cells can't produce enough nets. But actually, recently I also made some studies in the plasma of in the blood of patients with multiply myeloma, and we found that these patients has decreased Zincas level, but also the amount of neutrophil extracellular traps markers were decreased. So first we found that the exert of zinc can affect net release, but in some situation it can be totally invert situation. So I think this topic still needs some investigation.
Okay, we have one question more. Are any drugs known to stop making nets?
Yes, absolutely. Some antibiotics can stop making nets. For example, if I remember well, because we did this study in 2016, so 5 years ago, we studied the gentamicin effect and chloramphenicol effect, and gentamicin decreased the level of nets produced. So if we have the patients who is predisposed to autoimmune disease, and we can choose between antibiotics, we can choose the one which decrease the level of produced nets, because we don't want these patients to develop autoimmune disease, for example. So that, yes, that there are some drugs that affect net release.
Well, perfect. And what methods are used to measure nets?
The nets may be measured in vitro and in vivo. In vivo, it's a very, very difficult situation, because we have to use this microscope that can analyze the blood flow. In the ex vivo studies, we use the fluorometry, because the main part of the nets, this is the DNA, so we can bind, for example, cytox, green, or host to measure the amount of released DNA. We can also use the immunochemistry staining. So we use typical antibodies or specific antibodies against myeloperoxidase, neutrophil elastase, or other proteins, and the stain for DNA. And we can count the amount of neutrophil extracellular traps that are released from the cells. So main methods are immunochemistry, fluorometry, but we can also measure in blood the markers of release of nets. And the most common markers that are tested now, these are the complexes of DNA or myeloperoxidase, or citrullinated histones alone.
Okay, perfect. And last question. Can net be formed in stored whole blood?
Yes, they were found in the stored whole blood. Moreover, they were found also in the red blood cell products and the granulocytes products. So yes, they were found in blood banks and these products.
Perfect. So thank you so much for your presentation. And if there is no more question, question time is finished. We have finished. I would like to thank all the speakers for their presentations and the audience for attending the webinar. Today we have learned that morphological examination of blood cells is a crucial process after hematological analysis. Most laboratories need to review many blood samples, and manual review is labor intensive and time consuming. In recent years, we have seen many important advances in the development of new automated analyzers, and Mindray has launched the new MC80 digital cell morphological analyzer. So I think that everyone has learned something new today. Well, the webinar ends here. Thanks again to Mindray for choosing me as your host. It has been a great pleasure for me, and see you soon. Thank you.
Thank you very much.
I can't hear you.
Then you are muted.
Thank you so much for Dr. Javier and Dr. Olga. And thank you everyone for joining us tonight. It would be nice for you to fill in the survey before you leave, and it will help us to improve. If you want the certificate, please leave your full name and the email address, and we will email to you. Okay. Thank you and have a good day or good night. Okay. Thank you, everyone. Bye-bye.
Bye-bye. Thank you.
Thank you. Dr. Anna.